Ah, zero G's and freeze-dried cheese. Looks like another beautiful day on the spaceship. In this sketch, we'll be taking a closer look at everything that goes in and out of the spaceship as we discuss the ins and outs of transport across the cell membrane. Let's get to it. The walls of the spaceship represent the cell membrane. The cell membrane is selectively permeable, or semi-permeable, which we've represented with this semi-truck and its permed driver trying to enter the spaceship. The cell's semi-permeable membrane selectively allows some materials in and out of the cell, while other materials cannot pass through. Now, let's take a closer look at how materials are allowed in and out of the cell. Molecules that are either small or nonpolar, or both, can diffuse straight through the membrane to enter or exit a cell. Like this nonpolar desert bird that's small enough to fit through that netting to pass in and out of the spaceship. Some examples of molecules that can diffuse include water and hormones. However, molecules that are large or polar or both cannot pass through that membrane by simple diffusion. Notice the polar scene on the truck? That thing is definitely too big and polar to get through the spaceship's netting. Note that polarity is actually more important than size when it comes to the ability to diffuse. Materials that can't diffuse may still be let in and out of the cell by transmembrane proteins called transporters. There are two major types of transporters: channels and carriers. Channels allow molecules to move along their concentration gradient via facilitated diffusion, while carriers use active transport and can move molecules against their concentration gradient. Transporters only allow specific molecules through the cell membrane, which is why the transport tunnel of the spaceship has a crew-only sign. No space monsters at this entrance. Transporters can open and close to carefully regulate the passage of molecules they carry. Membrane receptors are areas of the transporter that coordinate this by binding a specific ligand. When the correct ligand binds the receptor, it signals to the transporter to open or close. Just like the transport tunnel is opened and closed by binding specific crew members' hands. Materials that cannot pass through the cell membrane via diffusion or transporters may be able to move in and out of the cell through endo and exocytosis. Usually large molecules and liquids are involved here. Let's check it out. In endocytosis, the cell membrane pinches in to bring outside materials into the cell's cytoplasm. You can remember endo, enter. Endocytosis is initiated when the material for transport binds to receptors in the plasma membrane, just like this alien craft is binding to a receptor pad on the outside of the spaceship right next to the enter sign. The pinching in of the membrane is initiated and completed by vesicle-coating proteins inside the cell. The most notable of these is clathrin, so we've represented this process with a clathrin-shaped alien squeezing the space wall in. As the membrane pinches in, it forms a transport vesicle, called an endosome, which you can remember with this alien who's clearly headed straight for the end zone. There are a few fancy names for endocytosis based on whether fluids or large particles are being transported. When a liquid material is transported into a cell via endocytosis, the process is called pinocytosis, which we've represented with this saucy alien having a liquid lunch of Pinot Grigio and Pinot Noir. On the other hand, this giant space hot dog represents endocytosis with larger, solid matter like bacteria. This process is called phagocytosis, and phago means eat, which explains why this alien is absolutely stoked to chow down. And speaking of space dogs, we've got a much less tasty but all the more endearing space pup exiting the station. Their exit represents exocytosis, which is the opposite process to endocytosis. In exocytosis, transport vesicles inside a cell merge with the cell membrane, releasing the contents of that vesicle outside of the cell. Remember, exo, exit. Looks like they're off to better things, like finding a utopia light years away where they'll get dressed in little sweaters and have their face needle-pointed on all the throw pillows. But I digress. Let's review. The cell membrane is a semi-permeable membrane that typically allows small or nonpolar molecules to diffuse freely in and out of the cell. On the other hand, transporters, like channels and carriers, allow some larger molecules to enter and exit. Materials that are too large to pass through the cell membrane by diffusion or transporters can enter a cell through endocytosis. This is initiated by the material binding to the outside of the cell, then proteins like clathrin coat the area of the cell membrane, causing it to pinch in and form an endosome. Pinocytosis brings in liquids, and phagocytosis brings in large solids like bacteria. Finally, exocytosis is the opposite process. It allows endosomes to merge with the cell membrane so large materials can leave the cell. All right, that just about covers the ins and outs of transport across the cell membrane. Now, if only we can master transport into the spaceship as easily, maybe I could be snacking on one of those foot-long dogs instead of this freeze-dried cheese. You better get out of here before you see what the chili looks like. See you next time.